This is a pre-lab video about before the electrophoresis, when we applied the DNA fingerprinting. The same name for fingerprinting is DNA profiling or genetic fingerprint. It's important when we want to just find out about individuals, how they relate to each other, or in crime scenes. So it just got figured out in 1984, the technique, so it's pretty new. Well, we can just say it. Now, the DNA is a long linear sequence and contains genes. We know that these genes are coding for proteins, and the proteins give us our unique phenotypes. So we have the genes, but between the genes we have segments that before that we didn't know, and still we don't really exactly know what they do, and we call them junk DNA between the genes. Now, it turned out that this DNA is not really junk. It's highly variable sequence that controls gene expression. So 99.9% of the DNA is identical for the same within the same species, but we have the DNA segment between the genes that is highly variable. So it's unique pretty much for every organism in a species. This DNA segment between the genes is called the STR, the short tandem repeat section, that it's made out of 4 nucleotides repeated over and over and over and over again in that segment. For example, GATA for 20 times, or TCTA for 30 times or 32 times. So it's kind of a long repeat. That's why it's called short tandems. The tandems are the 4 nucleotides and repeats. Now, let's go and check the example. Person 1, linear DNA. We have 3 genes on the DNA that they code for a protein, for different proteins, and between the blue and the green genes we have STRs. For example, we have 3 STRs over there. So the length of the STR is 3 times 4 is 12 nucleotides. Now, the STR between the green and the red might be for that person is 4 nucleotide, so 4 STRs we have. Now, the person 2 have the same gene, has genes at the same regions. They can be a little bit different based on the phenotype. What allele, actually not based on phenotype, what allele they just code for. Between the blue and the green, in this case, we might just have 8 STRs. And between the green and the red genes, we have only 2 STRs. So they're different in size, and we can use electrophoresis to separate the STRs, the different STRs, by size. So get rid of the other stuff besides the STRs with the help of restriction enzymes. So we can use enzymes to cut those STRs out. Okay, and the highlighted area is going to be used in our fingerprinting stuff. So cut out the STRs out of the DNA. Second step, amplify the STR segments in a lab with, we are not getting into it, but we make more and more because one is not enough for us. And we use electrophoresis to separate these segments. Electrophoresis, that's what we are going to do in a lab. The electrophoresis is using electricity to separate the different size of fragments, DNA segments, the STRs. So this is the electrophoresis. It has wells at the top, and we place the DNA from each people's DNA into the wells, and we put under electricity, negative to positive, the DNA by itself is negative, so it's going to migrate towards the positive sign. This is the whole thing is the agarose gel. And by the size, based on the size of the STRs, the smaller ones can move further from the well towards the positive charge, and the longer STRs cannot move that far away from the well. That's how we are going to separate those segments. See you in class.